Yesterday we talked about carbohydrates, and somebody remind me, did we get into polysaccharides in this class? Yes. So we're up to we're at have we gotten to this picture yet? Yeah. Yes. Okay. We'll just stop there. All right. So we were on starch and cellulose. Starch. Remember, for polysaccharides, we can clump them into two different categories based upon their function. And so there was storage polysaccharides, and there are structural polysaccharides. We talked about two storage polysaccharides. Who can tell me the two one storage polysaccharide? Jacob.
Starch.
Starch. All right, so starch is found in what kind of organisms?
Plants.
Plants, right? And when we say that they're storage polysaccharides, what are they storing? You're storing what? Energy. It's really long chains of glucose molecules, and glucose molecules our cells break down to use for energy to make ATP, and so that's what we're really storing. So we have starch in plants, and we have another storage molecule in animals. What is that? Glycogen. And glycogen is also a chain of glucose molecules, just like starch, except it's more highly branched and more complex. And for us, where do we store that glycogen at? So where does all of our excess glucose go from our blood? We store in the liver and the muscle cells. And so, then, segueing to what we left off with yesterday, we started talking about structural polysaccharides. So they have a different purpose. And cellulose is one of those structural polysaccharides in plants. So plants have both starch and cellulose. So in this class, did I talk about how these are both in plants, but they have different, did I write that here? No. All right, so let's look here. So cellulose and starch. What they both have in common is that they're both chains of glucose molecules, and they're both in plants. But what makes them different, and why they're called different molecules and therefore have different functions, storage versus structural, is because of what they're made out of. So starch is, yes, it's a chain of glucose molecules, but they're made of alpha glucose molecules. And remember, we talked yesterday about the two different forms of glucose rings, alpha and beta. The only difference between them is the location of these hydroxyl groups, one coming off the bottom, one coming off the top. So this is a chain of these glucose molecules. Cellulose is a chain of beta glucose molecules, the beta glucose molecules up here. And that difference in the chain of which ones there is the monomer of each one of these give them different functions. So as a result of being alpha glucose, these guys are involved in storage and energy. And as a result of being a chain of beta glucose molecules, this gives it the function of being structural. So let's talk about why it makes a difference being a chain of alpha versus beta glucose molecules, and why it changes the function. It has to do with the structure and the way that the glucoses bond together. Move this over here so that it'll record. All right, so let's talk about starch here. So starch is a chain of alpha glucose molecules. So I'm just going to draw the ring like this and put the parts on the end. So there's an OH and an H on this side, and an OH and an H on this side. I'm just going to draw those parts because those are the parts of the molecule that are important in the monomers bonding together, the glucose molecules bonding together. So this is the alpha. The textbook wrote an A like this here. The actual symbol for alpha is this, all right? And so, so this is your alpha glucose molecule, and I'm going to draw another one here. next to it. So I'm going to put here a OH and a H, OH and H. All right, so these are your two alpha glucose molecules. To hook them together, what needs to happen? Do you guys remember? What's the process that needs to happen to hook them together? Dehydration reaction. So therefore an OH comes off of one and an H comes off of another. If you recall back when we talked about that in this slide, I'm going to just go back real quick. We hooked here two glucoses together. Notice that it's the whole hydroxyl group off of one and the H off of the hydroxyl group of the next one. And let's go back then to this. And so in this case here, OH comes off, H comes off here, water molecule comes off, and this carbon now will bond to this oxygen, and that's this bond right here, all right, that we see right here. So every time then the next alpha glucose molecule comes, and so then if I were to draw another one, the OH and the H come off, and so on, and they hook together. And so that's your starch. How is cellulose different? If you just look at cellulose, do you notice how the oxygens are on top, bottom, top, bottom? Do you see that fluctuation there? Let's talk about why that is. It has to do with the construction of the beta glucoses. So that's starch. So let's look at cellulose. So when we make cellulose, we use the beta glucose. So I'm going to draw the formula here. So it's similar to the alpha on the left-hand side. But remember, what's different between the alpha and the beta is the flip-flopping of that hydroxyl group. So the hydroxyl group comes off the top here. And so if I draw another beta glucose molecule, this is your beta glucose molecule, another one here, OH, H, OH. So now we have to hook these two together. We have a problem here because when they come together, do you notice that the hydroxyl group is up on top here and this hydroxyl group is at the bottom? They don't meet up, all right? That's a problem. So in order to hook the two beta molecules together, we have to take this molecule, and it's like as we pick it up and flip it over, all right? So if we picked it up and flipped it, do you see how this hydroxyl group would be on the top now? All right? So therefore, if we flipped it, then what that molecule would look like, all right, so we need to flip upside down to make it work, to make these guys bond together. So if I were to pick this up and flip it, this hydroxyl group is now on top, and that OH was now on bottom. And then I flipped it over here, now I have an OH here and an H here. So this is flipped upside down from this one. Does this make sense? Okay. So now do you see that these two here match up? And OH can come off of one, and H comes off of another. You get H2O. This bond forms here to that carbon to the oxygen here, and that's this bond here, and that's why you have the oxygen up like that, because this one had to be flipped upside down to make it work. Now let me show you here what happens with the third one in this. So if I draw another beta glucose like this, and notice I drew it just like how it was. I didn't flip it upside down. Do you see now that the hydroxyl groups add up or match up? And so then you lose an OH and an H, lose a water molecule, and this bonds to that, and that's why it comes down like this. And then the next one, we'd have the same problem. The OH is on the top, so we'd have to flip it upside down, and then we get the fluctuating down, up, down, up. So every other beta glucose molecule you have to flip upside down to make it work.
Why do you have to flip it? Because before it was OHH, so wouldn't that just make a water molecule?
Yeah, the enzyme that helps to do this is very specific in shape, and it has to be two hydroxyl groups. You can't take an OH and an H. It has to be from the two from a hydroxyl.
Because there has to be remaining O, right?
That's right. All right. To make it, yes. To make it. All right. So that's a good question. All right, so therefore cellulose, this little change in structure changes the way it bonds, changes the structure of it, which then ultimately really changes the function of it, structural versus storage. And in fact, when we eat starch, our enzymes in our digestive system can actually break these bonds. Our enzymes can break these bonds when we eat starch, and we can then break it down into glucose and use starch for energy. We can use that glucose for energy. When we eat cellulose, that slight change in structure due to that hydroxyl group being changed, when we eat cellulose, which is in the cell walls of all the plant material that we eat, we can't digest it. Our enzymes do not digest that. So therefore, these bonds here, they can't break it down. And so we don't digest cellulose. It goes right through our digestive system, and we excrete it out through our solid waste. Sometimes they call that fiber. You've probably heard of fiber in your diet. Fiber is a good thing, even though we can't digest it, because it helps, adds bulk to our solid waste and helps it to move through our intestines and keep us regular, and so on. And so cellulose is a good thing, even though we do not digest it. But that slight change in structure is a big deal. All right, so the polymers with alpha glucose molecules are helical in shape. So starch was helical. Beta glucose molecules are straight, so we have a different structure there. In straight structures, hydrogen atoms on one strand can bond with hydroxyl groups on other strands. I'll show you a picture of that. It helps to add to its strength. And these parallel cellulose molecules held together this way are grouped into what we call microfibrils, which form strong building materials for plants, which is what this picture here is showing you. So here you're seeing a plant material, a leaf. Microscopically, that leaf is made out of many plant cells. These are the plant cells. The darker portion around here represents the cell wall. So this little circle means we're taking that circle and enlarging it. This is an actual real electron microscope picture of what the cell wall looks like. It looks like a bunch of fibers, doesn't it? Kind of crisscross and weave. Much like if you look very closely at a cotton t-shirt. It's the same thing with threads of cotton. And so it adds strength to the cell wall. When you actually look at one of these threads, each one of these threads is called a microfibril. Fibril is referring to like a fiber, micro meaning small. So they're like small little fibers that are crisscrossed and so on. But each one of these microfibrils is made from many cellulose molecules. So here is one cellulose molecule, a chain of those beta glucose molecules. Here's another one, and so on and so forth. And when they run parallel to one another in this microfibril, they can bond to one another. And so these little dots are hydrogen bonding going on between the adjacent cellulose molecules. So that also adds strength. So you have all these cellulose molecules bonding to one another and then coming together to form the fibrils, which then crisscross and so on to add strength and stability to the cell wall of plants, because plants do not have skeletons like us, and so the plant cells have to have more structure support, all right, around it. All right, and so that's your cellulose. And so we're going to write here a little bit about what I just talked about with the structure and digestion and so on. Enzymes that digest starch by hydrolyzing. This word, hydrolyzing, comes from the word hydrolysis. Hydrolysis is using water to split. So hydrolyzing, that's another way to say digesting. All right, or breaking apart. The alpha linkages can't hydrolyze beta linkages in cellulose. So as I said, cellulose in humans passes right through the digestive tract as insoluble fiber. So it doesn't dissolve. There are some microbes meaning bacteria, that have enzymes to digest cellulose. And many herbivores rely on them, like cows and termites, actually have microorganisms, bacteria in their gut, that help them to digest the plant material that they eat. So a symbiotic relationship, I use that word. Do you guys know what a symbiotic relationship means?
Isn't it when they like work together and it like there's like a positive for both animals?
Yeah, absolutely. So in this case here, you have the bacteria inside the organism. It's having a nice place to live. The organism is providing food for it. And at the same time, the cows and so on, the microbes are helping to digest some of that cellulose for them, and they can, as they excrete these enzymes, and they can use some of the glucose and the cellulose for energy. They both, and so here's the bacteria in the gut of the cow. The last structural polysaccharide is called chitin. And this is found in the exoskeleton of arthropods. We're going to see like some insects and things like that have, exo means on the outside of the, exoskeleton is on the outside of the body. We have endoskeletons that's in the inside of our body, exo is outside. Chitin also is found in the cell walls of fungi. And it's very strong, in fact strong enough that we have used it for surgical thread, all right, and surgeries. So, in the exoskeleton of arthropods, so this is a cicada. You may have seen a cicada. This is the exoskeleton of the cicada. It's shedding its exoskeleton, so it's crawling out of it. All right, it will grow a new exoskeleton, and sometimes you'll see on trees and stuff the leftover exoskeleton of the insect. And so this is made out of chitin. Chitin, the monomer of chitin, this is it. The difference and what's unique about chitin is that it has nitrogen. It's like glucose, all the other things we talked about is glucose. Glucose doesn't have nitrogen, so that's unique for chitin. And then this is just an example of using it for surgical thread. I don't have a picture of a fungi because the other category here was that it is in the cell walls of fungi. Fungi, like mushrooms, are in the kingdom of fungi. Mushrooms are very different than plants. So we have a plant kingdom and we have a kingdom of fungus, and so they both have cell walls. But the cell walls are very different. The plants are made out of cellulose. Fungus is made out of chitin, and there's many other differences between fungus and plants that we'll talk about later on in the course. So, but that's one similarity is that they both have cell walls, but they're made out of different things. All right, and that is carbohydrates. All right, so for lipids, actually before we go on to lipids, let's do a quick little overview of all the things that we talked about with carbohydrates. I did this in the first hour. Help to make a visual map. I'm going to do just a quick little thing with you, a little concept map of all the classes of carbohydrates and their relationship to one another. And so just to kind of give you, like I said, a mental map of the notes. Because sometimes I feel like, especially in this chapter, there's a lot of material and students get lost in the material. So I'm just going to, on a blank sheet of paper, we're going to make a little map about carbohydrates. So yeah, what we've done. So it's just a little, another way to look at the material and their relationship. So carbs. We can break down carbs into 3 categories based upon what? What would make sense to break them down into 3 categories? I would think it would be easiest to break them down based upon size. So smallest would be what? Monosaccharide, right? And as we go through this, again, self-assess. As we go through and I ask questions about things, if you don't know what you would put underneath there or don't know that, then that tells you that you should be studying. This is something that you should be able to sit down and just read along, all right? Just like that input later. All right, so disaccharides is next. And then what? Polysaccharides, right? So monosaccharides, hopefully we know what that means. Mono, one saccharide sugar. So we have one sugar here. this is the monomer of all other carbohydrates, right? So monomer of all carbs is a monosaccharide. Monosaccharides, they're used as what? What's the purpose? What's the function? Fuel. They're used as fuel. And the second function is to do with a little bit of the polysaccharides. They could also be building blocks for structure. What would I be looking for if I were given either a picture of a monosaccharide or a formula for monosaccharide? You guys remember the general formula for what a monosaccharide is?
No, I was about to say, I'm like thinking about it. I know it's like, it's ratio 1:1 to 2, something like that.
1 to 2 to 1. All right, yep. If we write it in the order, where they have this, CH2O. They have that formula here where they have twice as many hydrogen as oxygen or carbon. And so let's give an example of that. So what would be an example of a monosaccharide? Yeah, glucose, which is C6H12O6, an example of the twice as many hydrogens as oxygen or carbon. All right, so those are your monosaccharides. There are other monosaccharides besides glucose. There's fructose, lactose, but that's an example of one that you need to know. All right, so then your disaccharides. Your disaccharides, di meaning two sugars, right? Two sugars made from what process? How do you hook them together? Dehydration. A dehydration reaction. So what does that mean? They lose a water molecule. So disaccharides, an example. Anybody think of an example? Sucrose is a glucose put to, fructose. These are two monosaccharides hooked together, and the bond between the two monosaccharides, everybody remember there's a special name for that? Let's go G. G-L-Y.
Glycosidic linkage.
Yeah, glycosidic linkage. And this was made by dehydration, so we can say here that a water molecule came off. All right, OH from one and an H from another. So to use disaccharides, we have to break down the disaccharide into its respective monomers. So when we get table sugar, our enzymes in our digestive system break it down into glucose and fructose, and those molecules are small enough to get into our cells, and we can use it for energy, for building blocks, and so on. All right, so a disaccharide is too big to get into cells, so they have to be broken down first. All right, and then our last category is the polysaccharides. Now, I like to break, we in our notes broke down polysaccharides into two categories. What were the two categories based upon? The two categories of we did by function first. So we could do storage and structural. Is there another way you could do this to make your, could we do We talked about some in plants and some in animals. Could you do that? In plants and animals, you can do that as well. So this is not the end-all, be-all, way to make a concept map. I'm just kind of following what we did in our notes to kind of give you more of a visual here. So for storage polysaccharides, there were two storage polysaccharides. They were starch and glycogen, right? And for structural, there were two. They were cellulose and chitin. All right, so we could write out underneath here kind of some basic things. Do you guys want me to do that with you? Or is that helpful? Okay, so starch, we could write, and I'm going to run out of room in my, so instead of branching out, I'm just going to put numbers and write things about starch underneath it. So I'm afraid I'm going to run out of room width-wise. All right, so starch, we could write where they're located. Where are they located? Plants, right? Storage and plants. What else did we learn about starch that we could write? What does it make from? What's the monomer we could ask of starch? Monomer of starch. And we could say glucose, but let's be specific. Starch was what kind? Alpha glucose molecules, all right? That's what makes a difference between starch and cellulose. So these guys made of alpha glucose molecules. I'm just going to abbreviate glucose as GLU, glucose monomers. When we say that they're storage, I already asked this question earlier, but what are they storing? Yeah, they're storing glucose for later use, for energy. So they're storing glucose for energy for later. Anything else we can put for starch? Can we digest starch? Yes, we can digest starch, so humans can digest that. All right. So that's your starch. Glycogen is not found in plants. Glycogen is found in animals, right? Anything else about glycogen? I'm going to write here that structure-wise, comparing starch and glycogen, glycogen is also, we didn't really discuss this, but it's also made of the same alpha glucose molecules. So the third, this is the same. I'm just going to put the same from over here. We're still storing glucose for energy for later, so that's the purpose of it. And then the fourth thing, I'm going to put, comparing it to starch, it's more branched and complex than starch. And in our pictures in the notes, that's what we looked at here. The picture shows that. All right, so those are our two storage polysaccharides. Then we have, going over here to structural. Structural is found, or the cellulose is found where? Plants. In plants. And I'm going to put here more specifically cell walls. That's where you would find it in the plants, and that's what gives it structural support. Plants also have starch, but cellulose, they're both chains of glucose, but what's different about the glucose monomer? These guys are beta, right? So these are made of beta glucose monomers. Alright. And these guys, cellulose strands come together. They can hydrogen bond together to form, what were those fibers called? Microfibrils. So these guys can, I'll just put hydrogen bond to form microfibrils in the cell wall. And then remember the microfibrils, you know, are like those fibers that crisscross and weave and so on, and all of that together adds strength for the, and gives strength to the cell wall for the plant. All right. And can we digest cellulose? No. Humans cannot digest this. So this is our fiber. Moves through us. And lastly, we have chitin. This is found in two places. This is in, what do we call the outside skeleton? Exoskeleton. In the another one here, exoskeleton of arthropods, like that cicada bug. And where else? Where was the second place?
Fungus.
Yeah, the cell wall of the fungus. So it adds strength to that, and this is the only, what makes it different than all the other things is it has what element? Nitrogen. It has that group coming off the ring that has nitrogen, so that makes it unique. Alright? Okay, so that's kind of what I want to do, just to give you, kind of pull everything together so you can see how everything is related to one another. Is that helpful? Yes. Alright, yes. Alright, so in studying and things like that, you could try and do something like this on your own off the top of your head. Can you talk about polysaccharides conversationally like I did? Alright, so let's look at lipids. Alright, so lipids are our next macromolecule. And notice it says a diverse group of hydrophobic molecules. So hydrophobic, they're not dissolving in water. They're nonpolar, diverse. Just like carbohydrates have three classes, mono, di, and polysaccharides, lipids are also going to have three categories that we're going to talk about. They are fats, which are also called triglycerides, phospholipids, and steroids. So let's look at some basic overview of lipids and then look at each category. So lipids, they're large, hence the macromolecule, but they do not form polymers. So what does that mean? Unlike the carbohydrates, that the monomer was a single sugar, and all the other forms of carbohydrates were chains of those single sugars. Lipids, that's not the case. So they're large molecules that do not have monomers. Big thing about lipids is that they're hydrophobic, so they have little or no affinity for water. Why is that? Because their bonds in the majority of the molecules are nonpolar, so there's no charges. So when you put a lipid into water, the water molecules are more attracted to themselves than they are to one another. So they're hydrophobic because they're mostly hydrocarbons, which remember means that they're made of carbons and hydrogens. And those form nonpolar covalent bonds, so there's no charges. And so the three categories we're going to learn about are fats, phospholipids, and steroids. So let's look at fats first. There we go. So the fats are constructed from two smaller molecules called glycerol and fatty acids. Okay, so let's look at this name, glycerol and fatty acids. So glycerol, you may have never heard of the molecule glycerol before, but you should know something about it. Anybody have any idea how you would be able to know something about it having never heard of it before? Megan? It is the hydroxyl group. So how do we know that? Yeah, it ends in OL, which means that it's an alcohol, which means that it has the hydroxyl group, which is the OH group. So this is what I'm talking about. If you have not spent time memorizing those functional groups, we're building on, and so you still need to learn chapter 4, and we're building on to chapter 5 and so on. So you need to spend time to memorize those, all right? So these guys have the hydroxyl group, and that's how we know that. And then it's made from fatty acids. Fatty acid. What do we know about acids? What functional group does that have? What functional groups? So there's 3 people that know. Tristan? Carboxyl. So now carboxyl group, the next thing is, do you know what it looks like? So if you know the name, do you know what it looks like? Carboxyl group has a C with a double bonded O and an OH. All right, so from those names, that's the things that I should know, all right, right now about those. All right, so let's look at how fats are made from these 2 molecules here. So glycerol is a 3-carbon alcohol with a hydroxyl group, so that's what we just talked about, attached to each carbon. Fatty acid has the carboxyl group to a long carbon skeleton. So that's what this picture shows. Did I go too fast? No. Yes? Somebody say yes? And go back. Just gotta speak up. All right. So here we have, I know your box, I don't know if it shows up the light gray here, but this right here is your glycerol molecule. So we just wrote that it's 3 carbons with a hydroxyl group attached to each one. And so that's what we have here. And then the fatty acid group, fatty acid molecule has, here's the carboxyl group, see with the double bond and the OH. And then notice here as a carbon chain, and hydrogen's coming off of those carbons. And so that's the hydrocarbon chain. This part right here, those carbon to hydrogen bonds are non-polar bonds. And so that's what makes the fat not water soluble. So how we hook make a molecule of fat by hooking a glycerol to the fatty acid is by dehydration. So notice here, the hydrogen in the hydroxyl group comes off, water is formed, and now this oxygen binds to this carbon, and we lose 1 water molecule. Now, the fat, to make a full fat molecule, a fatty acid hooks onto each carbon here, or each hydroxyl group there. So really, the overview of a fat molecule, if I were to just do the generic, here's your glycerol molecule, and we just hooked 1 fatty acid here. Some of us can put Fa for fatty acid, and the second carbon would also have a fatty acid, and the 3rd carbon would also have a fatty acid. So to make a full fat molecule, we need to hook 2 more fatty acids to here. And so I always think of a fat molecule looking like the capital letter E, where glycerol is the backbone and the fatty acids come out, all right, from that. And so every time you hook a fatty acid to the glycerol molecule, what is lost? A water molecule, right? So to make one fat molecule, we lose 3 water molecules. One here, and one here, correct? All right? And so that's how you build a fat molecule. And so, so let's look at here. So fats separate from water, and because of those hydrocarbon chains here, they separate from water because water molecules form hydrogen bonds with each other and exclude fats. And that's because of the majority of the molecule of the fat are these fatty acid chains, which are hydrocarbon chains, which are nonpolar. So the majority of that molecule is nonpolar. So in a complete fat, as I just wrote here, that the 3 fatty acids are joined to that glycerol. They call those bonds that join them and what's called an ester linkage. And we call this a triacylglycerol, or sometimes a triglyceride. Tri meaning 3, because you have 3 fatty acids. Glyceride is coming from the glycerol. In layman's terms, we call that fat. Okay? And so this shows you a completed picture here of your completed fat molecule. So you have the 1 glycerol backbone, and notice here the little dots around here are the ester linkages. So this more specifically is the linkage that is formed between them. And so 3 water molecules were lost, and you can see that the majority of the molecule is hydrocarbon, and so therefore doesn't mix with water. Now, one thing about this is we're going to see that fat molecules, there's lots of different types of fat molecules, and one thing that makes one fat molecule different from the next is the characteristics of the fatty acids. Now this picture here shows you pretty much that the fatty acids have the same number of carbons, they're the same length, there's no double bonds, and so on, and so each one of these are the same. That doesn't necessarily have to be the case. There are different kinds of fatty acids. So how can a fatty acid differ? And that's the next bullet here. Fatty acids vary in length, so the number of carbons. So you can have a fatty acid that's 10 carbons long or a fatty acid that's 20 carbons long. And then also, if there's double bonds, the number and locations of double bonds between the carbon atoms. Now let's write that more specifically. The double bonds are between carbon atoms. That's what you're looking for. So, we have 2 categories of fatty acids, saturated and unsaturated. You've probably heard of these before. Saturated fatty acids, by definition, have the maximum number of hydrogen atoms possible and absolutely no double bonds. So that's why they call it saturated. When a sponge is saturated with water, what that means is that sponge is holding the maximum amount of water it can. You can't fit any more water. It can't hold any more water. It's saturated. So when we talk about a saturated fatty acid, when it has the maximum number of hydrogen bonds, or hydrogen, let's look at this as an example here. These fatty acids are saturated. There's no double bonds. Let me show you here what would happen. If we look at, let's say, this carbon here. So this carbon here is bonded, remember carbon can make 4 bonds. It's bonded once to the carbon behind it and the carbon in front of it. And so that makes 2 of the bonds. And so that means it has 2 more bonds, and those 2 bonds are bonded to each hydrogen atom. So this carbon, if it's in a carbon chain, can bind to a maximum of 2 hydrogens. Notice here, if I made a double bond here, let's say I made a double bond between these 2 carbon atoms. What's wrong with this picture? This is impossible. Why?
There's more than 4 bonds on the carbon.
Yeah, there's more than 4 bonds on the 2 carbons here that are made in between the double bonds, that are involved in the double bond. Each of them are making 5 bonds. So to make this correct, what do I have to do? I'd have to get rid of one of the hydrogens to keep the two of the hydrogens. So I have to get rid of this and this. And then you see that it would make the 4 bonds and be fine. But this molecule is not said then to be saturated with hydrogen, because you have the ability, and you can actually do this in the laboratory, where if you have a fatty acid with a double bond like this, we can actually break the double bond and add hydrogen to it. So you could break this bond and add the hydrogen to it, and now those carbons are said to be saturated with hydrogen. It has the maximum hydrogen that it can. You can't fit any more hydrogen on there because carbon only makes 4 bonds. And so that's the difference between saturated and unsaturated. So saturated will always have single bonds between the carbons and will have the maximum number of hydrogen, which in carbons in the center of it will be 2. The end carbon has 3 because it's to make that, it's not bonded to another carbon after that. All right? And so going back to this, unsaturated then, fatty acids have 1 or more double bonds. So all it takes is 1, but they can have more. And the major function of fats is energy storage. So fats that have saturated fatty acids, that are made from saturated fatty acids, we call in general saturated fats. And so like on food labels and things like that, they illustrate that and tell you how much saturated versus unsaturated fat. So that's what they're talking about. Most animal fats are saturated. So if the source is from an animal, they're saturated with no double bonds. There are some exceptions with that. One exception is fish oil. Fish oil is a fat that comes from fish, which is an animal, but is liquid at room temperature. So that's an exception to that rule that most animal fats, and that's why it says most, are saturated. Saturated fats are solid at room temperature. So think about bacon. You fry up some bacon. Bacon has a lot of fat in it, so you get a lot of fat in the pan, but you let that pan cool off, and what happens to that fat? It solidifies, all right? And so it's solid. And the key is at room temperature. All fat will melt and become liquid if you heat it up. And a diet rich in saturated fats actually contribute to cardiovascular disease, so through plaque deposits. And let me talk about why that is. So saturated fats, so it's a fat molecule that has the 3 fatty acids with no double bonds. So what happens is, let me draw it in a different way here. So if this is the glycerol molecule, and here are the 3 fatty acids coming down. When there's saturated fatty acids, meaning no double bonds, these chains, these hydrocarbon chains, are straight. So they go straight down. And so what happens is the next fat molecule, all right, can go right next to it, and the next fat molecule can go right next to it. And what happens is that they can pack together very closely and actually clump and form a solid. And so when you eat saturated fats and you have too much of those in your diet, as that saturated fat is floating in your bloodstream, it actually can adhere to your blood vessels and pack very close together, and they can actually form a deposit on your blood vessel and actually impede blood flow, and therefore it can be a problem, all right, with that. And so saturated fats are like that versus unsaturated, and we'll talk about unsaturated in just a second. Unsaturated fats do not have that same risk, and it has to do with the construction of them and the double bonds. So here's a butter, which is the animal fat, solid at room temperature, and this is just showing you here a saturated fatty acid. This is a specific type. It's straight. There are no double bonds in here. All right, so it's saturated with the maximum number of carbons. Versus unsaturated fatty acids. Fats made from unsaturated fatty acids are called unsaturated fats. These are better for you in your diet, and so when looking at food labels and so on, you'd rather have unsaturated fat than saturated fat. So examples, usually fats from plants, and then I talked about fish oils. Fish fats are usually unsaturated. Can somebody, well, let's do the next one and then I'll ask this question. And these plant fats and fish fats are liquid at room temperature, and we often call them oils. So we call it fish oil. Some people use like corn oil or vegetable oil. All right, so we have all these oils. Can somebody think of an exception for plant fats? Can anybody think of a plant fat that is not liquid at room temperature? It's not liquid at room temperature? Coconut oil is one, all right? So coconut oil is solid at room temperature. So that's another exception with that. So let's talk about why they're liquid at room temperature. And it has to do with saturated fats having double bonds. So this right here are unsaturated fats. So this is how saturated fats can kind of come together and clump together and be solid at room temperature. Solid molecules are closer together to make a solid. For unsaturated fats, this is the glycerol molecule. When they have double bonds, it changes the shape. Whenever there's a double bond, you get a kink. So the fatty acid kinks. All right, and so do you see that if you have molecules that kink out like this, that it's harder to pack them close together? All right, and so what we end up having is the molecules are spread farther apart, and therefore they're liquid at room temperature. They don't get close enough to be solid. All right, and that's, yeah, all right, and we are rounding out at the end of the hour. All right, what I'd like you to do real quick is to look at the packet I gave to you yesterday. I just wanted to do, tomorrow I'm going to give you some time in class to work on that, but I just wanted to do a quick intro to that. If you look at 1, 2, 3, 4, and the packet is pretty self-explanatory, but groups 3 and 4 in the packet, if you look at it, it talks about group 1, group 2, group 3, group 4, and group 5. We've talked about group 3 and 4. It's going to give you directions as far as what to do, questions to answer about those molecules. And the second packet that I gave you are pictures of a bunch of different molecules. It could be a carbohydrate, a lipid, a protein, and so on. And embedded in those questions, it's going to ask you to look through those pictures and identify certain molecules based upon what we've talked about. There are spots in there where once you identify it, you actually cut them out and tape them in. And so groups 3 and 4, I'd like you to look at and start today. You're going to get some time in class to work on it, and they're going to be due on Thursday.